And we have finished replication. We have finished gene expression, including transcription and translation. So you should feel really good with those. And now we need to look at a way of regulation. Do you remember we talked about in the metabolism chapter that like feedback inhibition and competitive inhibition and how you could regulate enzymes so that you could conserve energy? So that's one type of regulation, and that's based on protein to protein. Remember, like an allosteric molecule bound against the enzyme and the protein and changed shapes. But there is another way that organisms regulate their activity, and that is at the DNA level. Just shut the gene off. Don't allow transcription to occur. So that's one type of regulation. Now, prokaryotic cells have a specific form of regulation in that category where you can do that, and that is called an Operon structure. So that's what we're going to do next. We're going to learn at how prokaryotic organisms regulate their gene expression by shutting down transcription or turning it on when they need it. So turning it off and on, okay? And all of this is based on conserving energy, making sure that you don't make stuff you don't need, right? So, so that's what we're talking about next. Okay, so let's go to the PowerPoints. So genetic expression, we're regulating gene expression, and this is called an operon structure, and it's one way prokaryotic cells do that. Now, most of prokaryotic genes are always on. They're expressed at all times. You need them. They're going to need them all the time. But there are some times that there are genes that are not needed all the time. So this is the types of regulation we're talking about, those types of genes. Why would that happen? Well, there are some pathogens that only make a flagella when they get into our human intestines. Like they go through your digestive tract, through the stomach, and then once they get to the intestines, then they make a flagellum, or something like that, where they only need it at a certain time. So these are ways that we can do that. One way, there's another pathogen, the Pseudomonas aeruginosa, and it comes in and it kind of mimics, it kind of looks like a good guy, a bacteria that's not harmful, and our immune system doesn't attack it. And then all at once, when I have a whole bunch that have already replicated inside a host, they'll start producing toxins. And this is after quorum sensing. You remember when we talked about quorum sensing, and you can tell how many bacteria? It's a way the bacteria can determine the density of the population. So when you have a whole bunch of bacteria and this one knows it, it will turn on a gene and turn on production, and it produces this toxin, which if it had come in producing the toxin, that immune system, that macrophage, would have gotten rid of it and it wouldn't be a problem. But if I have a whole bunch of them and they all start producing toxins at one time, they can overwhelm the immune system. So it's a way that the pathogens can get in and kind of hide from the immune system and then do that. So that is an example of a way that they use operons to regulate. So let's learn about an operon and how this works. All right, so there's our gene. This is DNA, and we have a promoter. What happens at the promoter? That's right. This is our signal to start transcription. So this is where RNA polymerase binds to the DNA, and then it will start transcription. It'll continue until there's a terminator. That's not listed here, but it's understood. So this is a promoter to a terminator, and it makes mRNA from there. All right, and then between that promoter and terminator are our genes, and we know that prokaryotic cells can have polygenic regions. So this one actually makes 4 different genes. So this one mRNA strand is going to give rise to 4 different polypeptide sequences that'll form into 4 different proteins. Now, usually when you have polygenic regions, these proteins are related. They're all involved in the same thing. Maybe breaking down lactose. All of these produce enzymes that break down lactose. So usually in this polygenic structure, these genes are related. All right, so that's what you've got. Now in this particular thing, we also have another gene. So somewhere else down the DNA, there's a promoter and a terminator, and there's another gene that's nearby this region, and it's going to make its own mRNA. We refer to this as a regulatory gene. Because it's going to make an mRNA, and then it's going to get translated into a protein, and that protein is going to be involved in regulating this series of genes. So this is referred to as a regulatory gene. It's code for proteins that regulate the operon. How do they do that? They actually bind in this operator region. So there is again, these are just series of nucleotides, but it's a specific order that the protein made from this regulatory gene comes over here to the operator and binds, and it can either turn it on or turn it off, depending on what type of operon. All right, so those are the structures for that, this operator region. I think it referred to as like the traffic light. It's going to be able to turn it off or turn it on by controlling the movement of RNA polymerase. So that's something there. Okay, so let's keep going. There are two types of operons. We can classify operons as an inducible operon or a repressible operon. Inducible operons are usually off. They're not usually able to be transcribed. But if the conditions are favorable, the cell can turn it on and make it and allow transcription to occur. So an inducible operon is usually off, not transcribing, but you can induce it to come on. That contrasts with the other category, repressible operons. Repressible operons are usually transcribed. It's something the cell needs most of the time. So its normal configuration is on, so it's transcribed. But if it gets to a point where you don't need it to make any more of those proteins, it can be repressed or turned off. So repressible operons are always transcribed, but they can be deactivated, turned off, repressed by some proteins we call repressors.